Hello to our friends joining us via recording. We are reviewing for the unit number 3 exam. To help us review, we worked on some questions together in some groups. So we will go through and talk about some of these questions that we looked at. We will also be having a review session on Monday at 1 o'clock. So plan to attend that review session on Monday and bring your questions with you. So the first thing we're working on here are the different types of ion channels that are involved in the process of muscle contraction. Remember that muscle contraction happens through a couple of processes. So we talked about the biggest picture process that's called excitation, contraction, coupling, where we make the membrane change its charge to make contraction happen. That's big picture. Little picture, we also use that process called the cross-bridge cycle that tells us how myosin and actin interact with each other to make contraction happen. So I'm actually going to work my way through these statements from the bottom to the top because that's the way that it actually happens in a muscle when a muscle is contracting. So first thing that happens when a muscle is going to contract is it has to get a message telling it to contract. So here's where we tie together lesson number 9 and lesson number 10. Remember when we talked about neurons, that neurons spit out acetylcholine. That's one of their neurotransmitters that they use to talk. So one of the things that neurons talk to are muscle cells. When we talk about on the muscle, the kind of channel that's going to open because of this acetylcholine message from a neuron, what did we say was the specific kind of channel that opens up at the very beginning of excitation, contraction, coupling when we first attach to that acetylcholine? Yeah, exactly right. Kira mentioned chemically gated sodium channels. Chemically gated sodium channels. We know that we are chemically gated at the beginning of this process because we're opening in response to a chemical. We know that it's a sodium channel because sodium is what helps us to change the membrane charge. Hey, here's a wink, wink, nudge, nudge for us. When we are talking about the process of muscle contraction, never, never tell me that acetylcholine goes inside a muscle cell. It's the same deal when you're talking about neurons too. Never tell me that a neurotransmitter goes into a neuron. The only things that go into neurons when we're doing signaling, or the only things that go into a muscle cell, are the ions. My messages always stay on the outside and let charges come in. So at the beginning of a muscle contracting, a neuron spit out acetylcholine. That acetylcholine goes down, and it's the chemical key to open up a channel that lets sodium inside. Sodium comes in, my membrane charge goes up. But I don't just want my membrane charge to go up right where the neuron's talking to it. I need that charge to go all across the membrane. I need an action potential to take that charge everywhere. When we talk about action potentials, what's keeping an action potential going is the fact that at the very beginning, we let some sodium inside. We let some sodium inside, and that caused our membrane charge to go up. So when we're talking about action potentials moving, they're moving because of charge differences. When I talk about the kind of channel that changes because of charge, what kind of gate is charge sensitive? Who's opening and closing because of charge? Yeah, exactly. It's those voltage-gated channels. Voltage-gated channels. Now, in particular, when we talk about an action potential, there are 2 different kinds of ions that are involved in an action potential. The first one is sodium. Just like we open the chemically gated sodium channels to make the membrane charge go up, we open a voltage gated sodium channel to make the charge go up as well. But when the charge goes up, it can't stay up forever. To help us move our action potential across the membrane, we get high with sodium and then we get back down low with potassium. So the two kinds of ion channels that are involved in action potentials, moving a high charge across the membrane, whether we're talking about the T tubules in a muscle cell or whether we're talking about an axon in a neuron. Action potentials are always caused by voltage-gated sodium channels and voltage-gated potassium channels. Now, in particular, when we're inside a muscle cell, we have a special kind of channel at the end of the road. This is kind of the end of the excitation part of excitation, contraction, coupling. The end of the road for an action potential is when something gets pressed open or we push on something causing it to open. What's the kind of gate that means I push it open? Which kind of gate is pushed open? Yeah, exactly. Mechanically gated channels are pushed open. So at the very end of the road, when I've sent my message, my membrane charge went up, that's what I would call I get to the end of the road and I need to push something open. I'm going to use a mechanically gated channel that's pushed by somebody else who's sensitive to charge. So I push open my channels, and my channels are found on the T tubule, or they're in the T tubule. We press on them and they open up something in the sarcoplasmic reticulum. What does the sarcoplasmic reticulum store for a muscle cell? What's stored inside the sarcoplasmic reticulum? Yeah, exactly. Calcium. Calcium. The end of the road for muscle contraction, my ultimate goal is to spit out calcium. The way I spit out calcium is by pushing open or pressing open a channel on the sarcoplasmic reticulum. So friendly reminder, when we talked about muscle contraction, remember that muscles had chemically gated channels at the beginning, voltage gated channels in the middle, and at the very end, for good measure, we throw in some mechanically gated channels as well. The three kinds of channels for muscle contraction. We have 4 different proteins that are involved in muscle contraction. When we talk about our protein that actually moves, or the one that actually gets pulled on in the process of muscle contraction, who's actually moving when a muscle contracts? Who slides around? Yeah, exactly. Actin is my protein that slides around. Oh, I like that. Audrey said that actin is acting. It's doing the movements. It's making something happen. I love it. Actin slides. It moves around. The next protein on our list is the one that calcium attaches to. What protein in particular does calcium attach to? When it attaches to calcium, it changes its shape, right? When calcium is released. Yeah, exactly. So the protein that calcium attaches to is troponin. Troponin is my push pin. Remember that when calcium attaches to the push pin pops out, and we can no longer cover up our myosin binding sites. Who's the protein that normally covers up the myosin binding sites? Who's usually playing a trick on myosin? Yeah, exactly. Tropomyosin. Tropomyosin is all tricky. It's my protein that covers up the myosin binding sites. But it's a wet spaghetti noodle, right? So I've got to have troponin, got to have my push pin to hold that noodle in place. If calcium attaches to troponin, it pops off, and myosin falls out of place, or tropomyosin falls out of place, which means that myosin can attach to and pull on actin. By process of elimination, we can see that myosin is the protein that actually breaks down ATP. I chatted with one of my groups about this, and I'll draw a quick sketch for us here. Remember that when we talked about the myosin protein, it had those little golf club heads on it, right? So the golf club heads have the part where they're going to attach to actin, but they also have a part on them kind of toward the back side that we call their ATP Ace. ATP Ace. This is an enzyme that its job is to chop up ATP. When I chop up ATP back here, that allows me to change the shape of myosin. And when I change the shape of myosin, that gets me ready to do muscle contraction again. A couple of, or at least this statement here, is a non-traditional way to think about myosin. Here's a wink, wink, nudge, nudge. When you're studying, if you can think of a non-traditional way to describe what a protein does or what it looks like, or characteristics of it that maybe aren't the main thing about it, but they make sense, that will help you to review a little bit more thoroughly. So we don't think of, the first thing we think of with myosin is not necessarily that myosin breaks down ATP. We're maybe more likely to think about, oh, this is what attaches to actin. This is what pulls on actin. But the other thing it does is it uses that ATP to change its shape, to get the energy that it needs to stand back up and do contraction. So a non-traditional look at myosin protein. We were really awesome in our groups talking about these types of neurotransmitters here. I love that both of my groups made salty bananas, so we'll make one together too. Here comes my salty banana cell up here. When we talk about the salty banana cell, there are 3 ions that we care about: sodium, potassium, and chloride. Sodium, potassium, and chloride. Who do we find inside? Who lives inside the salty banana? Yep, exactly. Potassium lives on the inside. Absolutely. Which means that my salt, my sodium, and my chloride lives on the outside, right? So when we think about these things, the way that they're going to move is down their concentration gradient, meaning I've got a bunch of sodium on the outside, sodium wants to come in. I've got a bunch of chloride on the outside, chloride wants to come in. I've got a bunch of potassium on the inside. Potassium wants to get out of there, not going to stay. When these ions move, they take their charges with them. So sodium has a positive charge. Sodium comes in and makes things more positive. Chloride has a negative charge. When chloride comes in, it's going to make things more negative. And potassium has a positive charge. Potassium, when it leaves, takes that positive charge with it and makes the inside more negative. Now let's put this in the terms that we mentioned before we split up into groups. When I have an excitatory neurotransmitter, what's the abbreviation that I use to describe the kind of change that it does in the membrane? If I have an excitatory neurotransmitter. Yeah, exactly. So excitatory neurotransmitters make things called EPSPs. Excitatory postsynaptic potentials. Does excitatory mean that we're going positive or negative? If we're excitatory, is that positive or negative? Yeah, exactly. When I am excitatory, That's going more positive. So the one ion that when it moved made things more positive was sodium. Sodium brought its positive charge inside. Because of that, when I open up a chemically gated channel for sodium, I'm going to get excited. I'm going to get more positive. That's going to make me want to talk to my neighbors. So dopamine is one example of a neurotransmitter, a chemical, that it attaches to a chemically gated sodium channel. When it attaches to the chemically gated sodium channel, the inside of the cell gets more positive. So when I call dopamine an excitatory neurotransmitter, here's the first thing you should think. First thing you should think is it's going to make me an EPSP. It's going to make my membrane charge go up. That's the first thing you should think, because it's an excitatory neurotransmitter. The second thing you should think is that it is going to open up chemically gated sodium channels. Chemically gated sodium channels, because that's the only kind that makes me more positive. So here is the name of a neurotransmitter. I might give you a neurotransmitter that you've never heard of before on the exam, but I might tell you that it's an excitatory neurotransmitter, or I might tell you that it opens sodium channels. Again, we don't care about the name, we just care about understanding that excitatory means I get more positive, and it means the way that I do that is by letting in sodium. We drew such a beautiful banana on here. I'm not going to flip over to the other slide. We're just going to work it together here on this slide. We also have inhibitory neurotransmitters. Inhibitory neurotransmitters. Was serotonin the example I gave you? I think that was the one that I gave you. Serotonin is an inhibitory neurotransmitter. All the same stuff we did with dopamine, we can do with this one too. So it's going to generate an IPSP, that's an inhibitory postsynaptic potential. That means my membrane charge is going to get more negative. And the types of channels that made me more negative were potassium leaving with its positive and chloride coming in with its negative. Hey, notice how we didn't have to know the name of our neurotransmitter to predict the answer to the question, right? It's the same deal like we talked about with stuff in the blood-brain barrier. You don't have to know what we're talking about. We just need to know the important information. And the important information is there no matter what we call it, what neurotransmitter we use. So, excitatory versus inhibitory neurotransmitters. It all comes back to that salty banana. Audrey had asked earlier if I was proud of my children for making salty bananas. Of course, I'm so proud of y'all. You're great with these salty bananas. I love it. Last thing I asked you to work on together was to review what the functions of the parts of the brain were. So let's work our way here through our list here, our last bit of review. First thing I asked you about was jet lag. Which part of your brain is malfunctioning when we have jet lag? Who's out of whack when we are experiencing jet lag? Yeah, exactly. So the pineal gland is problematic when we have jet lag. What is it that the pineal gland makes? And what is it making too much of at the wrong times when we have jet lag? Yeah, exactly. So this makes melatonin, and in jet lag, it's at the wrong times. The pineal gland makes melatonin, but when you are jet lagged, it's because you're spitting out melatonin at the wrong time of day. So yeah, we need to reset it with that bright light. Hopefully that helps us to spit it out only at the right times. When we talk about the part of the brain that's most inhibited by alcohol, what part of the brain did we talk about as being most inhibited by alcohol? Yeah, exactly. We talked about that with the cerebellum. Remember, our trick with the cerebellum, we had said that the job of the cerebellum is basically to not make you look drunk. So a good way to remember who gets influenced by alcohol is the cerebellum. If you look like a drunk person, if your movements are all funky, you don't have good balance, all of that is because your cerebellum has been inhibited by the alcohol. What was the part of the brain in particular that helped us with that fear emotion? Which part of the brain makes us feel scared? Yeah, exactly. The amygdala, or sometimes we call it the amygdaloid body. Same deal. So it's the part that makes you feel scared. It's the part that makes you feel angry, the amygdala. What was the specific part of the brain that helped us with skeletal muscle movement? What did we come up with for this? The specific part of the brain that does skeletal muscle movement. Yeah, I love it. Part of the brain called the precentral gyrus. Absolutely, precentral gyrus. Hey, which lobe of the brain is the precentral gyrus found in? Where's that one? Yeah, exactly. That's found in the frontal lobe. Precentral gyrus, helping you with moving your fingers. Other things the frontal lobe does. Remember, we talked about your critical reasoning, your decision making. All of that's the same kind of stuff that happens in the frontal lobe. When we talk about blocking unwanted movements, so blocking things like tremors or tics, what part of our brain helps us to block those unwanted movements? Who keeps those things at bay? Yeah, exactly. The basal nuclei. The basal nuclei. Remember, this is the part of the brain when someone has Parkinson's or when someone has Tourette's. This is the part of the brain that is not functioning, leading to those tremors or leading to those tics. And our last question here, asking about the parts, there are two of them, of your brain that regulate breathing. What are the two parts of the brain that do breathing? Yeah, so first one is the medulla oblongata. And I'll mention with the medulla oblongata, we call it the medullary, let's see if I can spell this, rhythmicity center. Helps us to set our breathing rate. And then we also have the pons. And the pons has something called the pontine respiratory center. Pontine respiratory center. Both of those help us to regulate the process of breathing. We are out of time. We're more than out of time. Thank you all for sticking with me here. As we wrap up today, are there any last minute questions about this content before we call it a day? And help me out in the chat too. What topics in particular do you want to spend time on Monday? What particular things do we want to make sure we cover? Okay, so Temiteo, lesson number 9, some of that muscle stuff. Okay. All right, well, while we are still thinking, I'm going to go ahead and end the recording. But remember, we're going to have a review session again on Monday at 1 o'clock. If you are free, please plan to stop by. Otherwise, best of luck on wrapping up your assignments and good luck on the exam.